Going live. Okay, live. G'day viewers. We've got Bill Andrews with us today, who's the author of Curing Aging, a book about telomere rejuvenation. Bill Andrews is also the CEO of Sierra Sciences that work on rejuvenation therapy, especially with regard to telomere health. And he was also the subject alongside Aubrey de Grey in the life extensionist film, The Immortalists. So, yeah, Bill, do you want to give us a little background on yourself?
On my science or on my personal self?
Both, hopefully.
Okay, well, I've lived all of my life in the United States, but born in Michigan, raised in Southern California, and graduate school in Georgia, and then started biotech in San Francisco Bay Area. And then in 1999, I started my own biotech company in Reno, Nevada. And I've been there, that's Sierra Sciences, with 100% focused on finding ways to lengthen telomeres to reverse aging and declining health. I'm personally an ultramarathon runner, but I've also competed in a lot of other sports too. I used to actually hold the world speed record for barefoot water skiing back in the early 70s, where I went like close to 80 miles per hour. But most of my, I've all other sports too, but the one that I've been most fond of is just running and venture running. So I tend to run 50, 100-mile races once a month. Just for the adventure. I've done races in the Himalayas at 18,000 feet elevation. I've done races in the lowest point in the United States, Death Valley, nonstop at 130 degrees, finishing at the highest point. Yeah, no, it's fun. There's
in the Himalayas where there's not much oxygen, you can fall off a cliff. That's certainly not. No, like these extreme sports. I guess it's not really classified as an extreme sport, but at that altitude, there's a bit of risk there, isn't there? Aren't you worried that you might accidentally hurt yourself?
Yeah, but I never get myself into those situations. Even in the Himalayas, I was mostly on roads or dirt trails, and never got myself exposed to a situation where I would be next to a cliff, unless I wanted to get next to a cliff, things like that. It's very easy to acclimate to altitude and heat and things like that. So as long as you do a lot of preparation beforehand, which is important if you really want to have fun doing it, they're great things. Now, of course, I wouldn't call my races in the Himalayas that much fun. Both times I've run that race twice. It's 138 miles nonstop at 18,000 feet elevation. And the first time I got a gallbladder attack at 50 miles, had nothing to do with the running, but I had to be airlifted out. So that was a bust, but I went back 2 years later and ran it. But this time at 97 miles, I got some really weird thing happening to me. And I thought maybe I was having a heart attack. And I was at the lowest point of the race, lowest altitude of the race. And I said I needed a doctor, and doctor came and checked me out, said my heart was fine. So I just got up and finished the race. But I later figured out that I was just having some kind of allergic reaction to something in the air that was in this small village that I was passing through at the time. Because as soon as I got out of that village, I felt absolutely fantastic and was able to run up another 18,000 foot mountain. I ran up from 11,000 to up to 18,000.
Wow, that's incredible.
Yeah.
So I mean, like, it seems as though like the sports that you're involved in, now you've said somewhere else that long distance running is somehow helps generate the telomere growth. I'm not sure if I got that correctly. Is that true? Do you believe that?
No, I've said that endurance sports, running, swimming, bicycling, walking, as long as you do it in a way that keeps it fun. If you're on your hands and knees crawling because you're out of energy and you're throwing up and stuff like that, that's not the right way to do it. But if you just approach these sports like they're fun backpacking trips with adventure, take a camera, take pictures, socialize with other runners, things like that, that's actually really good. You actually end up boosting your antioxidant levels so high that the overcome the the free radical levels that increase from the exercise, and so your net oxidative stress is less. And that has resulted, because free radicals is one of the causes of accelerated telomere shortening. The people that run all the time, like run almost every day, doing marathons and stuff like that all the time, they end up having much longer telomeres than their friends their same age that haven't been doing all this running. But it's not because their telomeres got lengthened. It's because they've reduced the rate of what I call accelerated telomere shortening due to inflammation and free radicals. Now, an example of that is that if you're an occasional runner, you go out and run maybe once every week or two, and then you sign up for a marathon, you'll do really good in the marathon, but and you'll be finished and feeling really like you had a really great day. But the next day, you're going to be stiff as a board from all the inflammation. Okay, that causes accelerated telomere shortening, that inflammation. But if you are the type of person that runs every day and you go out and run a marathon, you'll have a good day, but you won't have the inflammation the next day because your body's so used to it. And so as a result, it doesn't have the accelerated telomere shortening caused by the inflammation. So I'm a big believer in that. I've been a believer in it long before I even knew about the importance of inflammation and telomere length. When I first got into my endurance, at least the ultra marathon distances, I was surprised how many 80-year-olds there were that were competing and beating my pants off, and looking like they're teenagers. My very first 100-mile race, I was handheld by a 75-year-old woman. And when we got to 95 miles, she said, well, okay, Bill, looks like you got it. I'm going to run on ahead. And then she beat me by like 20 minutes.
Wow, that's incredible.
At 75.
Yeah, it's 75. Yeah, right. And did she look 75?
No, not at all. She was an incredible woman. She still is. And I think she's now in her 90s.
Yeah, well, I mean, like these are sort of lifestyle choices that people can make, I guess, in terms of like allowing them to increase the odds of being around when the science really does get that much more mature, such that people can, quite easily know what to do to scientifically extend their health span, right? What are some of the lifestyle choices that you'd suggest people, agnostic of their preferences, whether they like, long distance running or high intensity training, what do you suggest that people can do now to increase the odds of?
Well, first, I wouldn't encourage somebody to get into long distance sports, endurance sports, unless they liked it. And the way that I've made it myself like it is to combine it with adventure. So I'm always on a new trail, always exploring and things like that. But the other things that people can do, and walking is probably just as good as running because it burns just as many calories. Maybe it's even better. I don't know, but I'm impatient, so I can't just walk. But the other things you can do are the antioxidants, omega-3 fatty acids, vitamin D, and those have been published in scientifically peer-reviewed journals showing that people who've been taking those for a long time have longer telomeres than their friends that don't. Now, they don't lengthen telomeres; they just decrease the rate of the shortening. Smoking, obesity, both of those accelerate telomere shortening. So, quit smoking and lose weight. Depression, the other kind of stress, psychological stress, the kind that your boss gives you, those things cause accelerated telomere shortening. So you want to meditate, do things you can reduce that. Find solutions to depression. There's a lot of things you can do, but it's pretty much the happier you are, the longer your telomeres. So
how does depression and stress result in early, I guess, degeneration of the telomeres?
I don't know exactly, but I assume somehow it's increasing the oxidative stress and or inflammation. It surprised me to find that there's studies, there's several of them. There's like on the stress, people have looked at financial stress. They've looked at adults that were abused as children. They find that they have shorter telomeres than adults that their same age that weren't abused as children. There's just several studies now looking at that, and they all come up with the same thing. My research isn't to look at stuff like that. That's for me just reading other science and discussing. My research is totally focused on trying to lengthen telomeres. And I don't know if stress has nothing to do with telomere length. We'll probably find out when we have some way of lengthening them. You know, and then there's always cause and effect and stuff too.
Right, yes.
Telomeres causing the stress. I don't know.
Yeah, so correlation isn't causation. It's very hard unless you've actually got a strong hypothesis that's testable, and for it to go through the proper trials, which requires funding and a lot of effort. We're not quite there yet. So what sciences are you excited about then? What are you working on specifically first? And we can talk about others later.
What I was going to say is there's no way we're ever going to know if these things are about cause and effect until we have some way of lengthening telomeres and then ask what happens. Then we can say it's a cause or a result. But the thing that I'm doing, my research is totally focused on finding ways to lengthen telomeres in all of our cells. In every single cell of our body except our reproductive cells, beginning somewhere at the primordial germ cell place up until somewhere in the middle of spermatogenesis and oogenesis, we have telomerase produced in our cells, and that keeps our telomeres from shortening. And I think that's essential, because if we didn't have that happening during that short period of time, then our children will be born with shorter telomeres than we have, and their children will be born with shorter telomeres than they have, and we would be extinct as a species by now. So we have to have some way of preventing telomere shortening in the reproduction cycle, but it's, for some reason, and evolutionary biologists can explain it really well in terms of survival. Apparently it's better for the survival of the species if we knock off our old. And humans, dogs, cats, horses, sheep, pig, and deer all do it by causing their telomeres to start getting shorter as soon as they start becoming more than just a single cell. Whereas other animals like mice and all rodents and fruit flies and roundworms and etc., yeast, they do it by different mechanisms. So aging isn't, I think aging is kind of like a relatively new thing in the evolutionary cycle, because there's so many different ways to cause the aging process. Mice age by oxidative stress and mitochondrial dysfunction. I'm sure that's something we get affected by too, but not as much as our telomeres. Humans, you know, we can, as I said before, we can actually enhance our enzymes to prevent the free radicals. But the I was trying to come up with that word antioxidants there for a minute, but mice can't do it. You put a mouse on a treadmill, you're going to accelerate its aging tremendously. But it's because they have really high levels of oxidative stress and mitochondrial dysfunction. Now, if you smoke or you're obese, then yeah, oxidative stress plays a big role in your aging process. But if you don't, if you really lead a really good, healthy lifestyle and you've got really good genetics, there's no reason why you wouldn't live to be in your hundreds. And the only reason that you'd end up succumbing was because of your telomere shortening, unless you were run over by a bus or something like that. But so what I want to do is I want to lengthen the telomeres. We are mostly doing it by trying to find ways to induce expression of the enzyme telomerase,
Yes.
and which is an enzyme I led the research and the discovery of back in the early 1990s.
In humans, right? Elizabeth Blackburn discovered it, like in mice. Is that correct?
No, she discovered it in Tetrahymena, a single-cell pond scum organism,
Yes.
back in the mid-1980s. There's documentaries out talking about all that and stuff like that, so people can go to like Never Grow Old, or there's another one I can't remember that came out before the Immortalists that go through all that. So they found it in Tetrahymena. It turned out Tetrahymenas components for telomerase are very different from the human and the mouse. We ended up finding the mouse and the human, components right after we found the human components. But the yeah, I led the research to discover the human telomerase, and it was the first component that we discovered was called the RNA component, and that was so unlike the tetrahymena component. We just had to start from scratch, and I'll tell you, it was one of the toughest things that me and Dr. Bryant Villaponteau and Dr. Julian Fong and Dr. Walter Funk, a few others, all worked on it. I mean, I'll tell you, I don't think any of us will ever imagine doing something more difficult than that was. And it's the only project that I've ever been involved where we all got together and celebrated a 10-year reunion on discovering that enzyme. Yeah, so we discovered it. Now Sierra Sciences is trying to find ways to get that produced in all of our cells, because all of our cells do contain the gene for it. It's just shut off, and you'd have to ask an evolutionary biologist why that is. We have found things that turn it back on. We found both natural products and synthetic products. The synthetic products are a lot more potent than the natural products, but still anything's better than nothing. The natural products are a benefit to people because they don't require FDA approval, and so they're on the market already. But the synthetic products, they require FDA approval or whatever country, regulatory approval from whatever country you're in. Some of the synthetic products now, and telomerase inducers, have been now approved for use on skin, and so there are skin creams available now with that in it. But all that means nothing to me except a source of funding for our research, because what I really want to do is I want to make it so that we can find something potent enough to turn somebody like Betty White into a 24-year-old again.
Right. Yeah.
I can't wait for the day when she walks out on stage looking 24 again. And you know, do it for myself, my family, and but the mouse studies that was done in Ron DePinto's lab at Harvard before he became the head of MD Anderson showed pretty convincingly that lengthening telomeres does cause all kinds of age-related reversals. But as I said before, mice don't age by telomere shortening, so he had to engineer the mice first to make it so that they age by telomere shortening.
Okay. Was that by reducing oxidative load or susceptibility to oxidative load?
No, he didn't do that. The easiest way for me to describe it is that mice usually succumb from oxidative stress and mitochondrial dysfunction at about 3 years old. So what he did is he created mice that died from telomere shortening at 2 years old. And so it became their essentially shortest, the stick of dynamite with the shortest fuse.
Right,
and,
yeah, I see. Yeah.
So when these mice got really old looking, and I'll tell you, these mice did look different from other old mice. They looked more like humans that were aging. Male pattern baldness, hair color changes, all kinds of things. They lost the ability to reproduce again. They couldn't, their brain functions had deteriorated. But when Ron DePinto lengthened the telomeres in these mice by using 4-hydroxytamoxifen to induce the estrogen promoter, which he had put in front of the gene to regulate the gene expression. It won't work on other mice, and it won't work on humans. It only work on those mice. But he found that when he did lengthen the telomeres in those mice, they got younger in every way imaginable, including they could reproduce again, brain functions came back. I think there's a possibility that this might be one study among others that suggests that things like Alzheimer's and other forms of dementia might not be the loss of memory as opposed to the loss of access to the memory. So I believe telomeres has a good opportunity for actually reversing Alzheimer's. I just hope. But right now, of course, we try.
So at the moment, there are a couple of, like you mentioned, like some supplements before. Those are natural sort of ingredients. Product B and TA65 you've mentioned elsewhere. Do you take these yourself?
Yeah, every day.
Yeah.
Yeah. So TA65 does have clinical studies. A new one just came out showing that it actually has a beneficial effect of decreasing the onset of macular degeneration, which is one of the major blindness in the elderly. Product B doesn't have any clinical studies. I think they have some underway. that's produced by a company called Isogenics, and we discovered the natural products for them. Okay, so they created product B and put that on the market. And as for our services, we get a royalty, which we use to fund research. The skin cream products, now those are, I want to say 200 times more potent because they contain a synthetic chemical that we were able to do medicinal chemistry on to optimize, okay, to make it less toxic and more potent. Those are pretty potent, but they're not approved for oral use. We license it.
So what immediately comes to mind is, why don't people buy that and just swallow it anyway? I mean, off-label usage, right? I mean, people do that with other drugs.
Well, but this is a skin cream, and I was thinking, would you swallow your like suntan lotion? You know, I'd
rather package it first. But you know, I've heard people say you shouldn't be putting stuff on your skin which your body shouldn't, wouldn't be able to break down. Your skin is an organ, isn't it?
This might sound horrible, but that may or may not be true. But right now, we just discovered the chemical and licensed it to a company that turned it into a skin cream, and we get a royalty out of it. Which, if it's a bad thing to do, then I like to call it it's marching into hell, but for a heavenly cause. You know, I found our research to come up with some really potent and safe, FDA-approved pharmaceutical that's going to, reverse our aging and declining health, make us healthier. You know, as I said, just like in the mice studies with rondopinol. That's the only, we know exactly what to do. The obstacle is the funding, and I just hope more and more people buy the skin cream and the supplements to start contributing towards our funding.
Do you use the skin cream?
Yeah, I use the skin cream. I got to confess, skin cream is not my thing in general, so I forget to use it a lot. I don't wear lipstick either, you know. it's not my thing, but I'm more interested in the overall health, so I'm looking forward to a pill you can take, or you know, the other approach, and you might have heard this from Liz Parrish of BioViva, is doing the gene therapy approach. Deliver, instead of causing your own cells to induce telomerase expression from its own genes, deliver a new gene carried on a virus to all your cells, and then you produce telomerase inside your cells there. We are actually working quite close with Liz Parrish now, and have signed a deal with her company and another company to jointly work together to providing this therapy. Unfortunately, it's super expensive. So again, I'm just looking at this as another opportunity for us to get funding to make our affordable.
Well, we hope that those costs come down. But look, as you may know, I interviewed Liz Parrish about this recently, or actually last year, not that recently anymore. But yeah, she tested this on herself. Would you have you also had therapy yourself?
You know, I'm a chicken. Liz Parrish is very brave. I'm very impressed with her doing it. She did put her life at risk by doing it. How much, I don't know.
Certainly walks the talk. So
she's really determined to make something better. Her big interest is really children. She'll always be very open to saying that. She'd like to see better therapeutics and stuff like that to help children, but at the same time, she wants to see, she's sick and tired of seeing unhealthy people, old or young. And she and I both believe that lengthening telomeres is a good way to reverse those kind of things. And so I'm super impressed. People should keep an eye on her, you know, because I think exciting things are going to come from her. And I'm glad now that we are working with her in a joint deal, and we'll see what we can come up with.
So what's the nature of your joint deal with her? What are you doing? Are you supplying like scientific services? Are you doing the research for her?
Yeah, we're doing the, let's say maybe on more of an advisory role, the molecular biology that's involved, and we're setting up a what would be called a large-scale production facility to make the cost of producing the virus cheaper. Okay, so we aren't so much developing the virus as much as we are at providing a service to make it a large-scale production facility for her to be able to get the virus to treat people with. And it's going to be expensive for those people out there thinking right now that, hey, that's what I'm going to do. Right now, we're estimating the cost to be like just to produce enough virus for treating one person could be as much as $700,000. And so in order for the company to make a profit, people are going to have to pay like $2 million just for one treatment. Now, one treatment might be pretty wonderful for people, but it's not going to be the kind of thing everybody can do. And that's why I want to get my research company going to focus entirely on getting an affordable treatment that everybody can do. And that's going to happen because I own 100% of my company. So there's not going to be any like large pharma or somebody else that's going to pull the rope on me to stop me from doing this research.
How will the price go down? It seems like that's a pretty hefty fee. Now, some people can afford it, but at least in the short term, most people wouldn't be able to afford it. So how long will it take do you imagine to sort of get the price down for this therapy to be available for more people, and by extension having more samples to do more further research on?
Well, first, the way it's going to become affordable is by using an alternative to the gene therapy, and that's to use a small molecule pharmaceutical drug. Now, if we had all the funding we needed right now, we could, with almost complete certainty, have such a drug ready for testing in humans in less than a year from now. But the problem is the funding. Our burn rate before we lost our funding from the global financial crisis was about $1 million a month. And that's what we need to get back to right now. The products that we have on the market so far, we don't have on the market, but we've licensed other people to put on the market and we get a royalty from. That's earning us about $200,000 a month right now, which isn't enough to really do the research. But I look at this gene therapy approach that BioViva is going to do is a way to actually fund our research to come up with that small molecule. So once we start getting the royalties from that, then start to count down from a year after that, and we'll have that small molecule.
Right. So what would be the difference? If I mean, like, of course it would be cheaper, but is there still something you're missing out on by just taking a pill instead of doing gene therapy, do you believe? Or do you think these pharmaceuticals will do everything that the gene therapy does in the future?
Yeah, I think the pharmaceuticals, I mean, the goal is to be able to get that pharmaceutical into the blood and to be able, and then not have it be removed from the blood, so it gets to every cell in the body. And that's pretty much a function of medicinal chemistry. So we have really good medicinal chemists here. And so once we find drugs that work, then we can modify those drugs to make them so that they, let's say, survive the stomach and get into the blood. If they don't survive the stomach, we can inject them into. But then there's still other things that can happen inside where they could be destroyed in the blood. So that's all medicinal chemistry to overcome those problems, and that's going to be a lot easier than actually finding something that's potent enough. And when we extrapolated all the results we've had in the past, we can definitely see that we'll be at potent enough to stop telomere shortening, and then even after that, potent enough to reverse telomere shortening in a year from now. A year from when?
In 1 year now. Oh, yeah. Right, in a year from when you have it, right? So when do you think you'll
have the funding? Okay,
depending on.
So let's say we could have the funding within a year from the gene therapy stuff, or maybe if the skin cream or the supplements get a boost, and hopefully everybody get out there and buy it now. But let's say 2 years. Okay, let's say it takes a year for us to get the funding, then a year later we have it. So it's a year or two from now before we'll have a small molecule telomerase inducer drug that will be ready for testing in humans. When I say that, I mean ready for clinical studies.
Yeah, human trials. So you'll be testing on animals in the meantime, but in 2 years, depending on funding, you suspect that you'll have this ready for human trials, which will probably take, what, 3 years?
Well, we want to do preclinical studies with animals at the same time, but the problem is that there aren't, as I said, most animals don't age by telomere shortening, and it's hard to have a disease target when you're talking about something like aging, because you can't give somebody a drug and then 50 years later ask if they're still alive, because that would take 50 years to do the clinical study. But we are looking at the fact that people who are infected with the AIDS virus actually have accelerated telomere shortening in their immune cells, and that's why they get immune deficiency. That's acquired immune deficiency syndrome. And so turns out cats also suffer from AIDS, have the virus called feline immunodeficiency virus. And so we are gearing up to do our preclinical studies on cats, just to show that cats can live, who are infected with FIV, can live normal lives even though they're still infected with the virus, because their telomeres don't get short. So that would be, I would say, move forward pretty fast in clinical studies, so that in the FDA approval process, so that we could get into humans quite quickly. The side effect is that not only will they survive their AIDS, they will live longer. Now, unfortunately, in the United States, fortunately or unfortunately, depending on your perspective, there are really good treatments right now in the United States for treating people with AIDS. And so most people with AIDS do live pretty much normal lifespans, but third world countries and stuff don't. And so I look at this as an opportunity to help with the AIDS epidemics in some of the third world countries.
Oh, absolutely. That sounds like an awesome cause.
But
I really, I mean, often people get the impression that, like, a lot of these therapies are just going to be benefits for the ruling elite, and so believe that, and some people even go so far as saying, well, we shouldn't do the research because it's not fair. Some people get it and other people don't. There'll be power imbalances and all those sorts of things. So that aside, we can see an example of this research going to a third world country and helping people who can't afford other means, can't actually, who have AIDS in Africa, but can't afford to, I guess, maintain their health in other ways. So it seems like this approach would, I guess, attack AIDS at a fundamental level.
Yeah, and the side effect is they'd get younger and healthier. We've seen a lot of movies. We know all the kind of things that can happen with the cure for aging and things like that. And maybe they're all going to happen, but the bottom line is that when push comes to shove, the cure for aging is going to be a good thing. And 100 years from now, when we have a cure for aging and we have the elite, social elite, being able to take the drug and nobody else, the other problem is, I often say, is that young people will never be able to compete for jobs because the old people don't get old and they just keep getting better at their jobs. And so we're going to have problems like that. But I think if there was somebody who said we should put the cure for aging on the ballot 100 years from now, saying we should ban it, I don't think anybody's going to say, no, don't ban it. We'll find some other solution to the problems we have, because aging is a major problem we have right now. And most people, this whole crap, I'm going to call it, like aging gracefully and things like that. Well, yeah, sure, at the beginning stages, but boy, you go visit a assisted living home or a hospice or something like that, and I
used to work in one.
Yeah. Well, congratulations. I think that's really honorable of you, because I think that's going to be one of the #1 needed professions in the new years if we don't get a cure for aging within the next 10 to 15 years. Japan is the worst country for this, because they are going to have something like 35% of their population over 65 in 20 years from now. The United States is also going to have a problem like that, and we're going to have enough people to take care of them. Caregivers are going to be extremely important. And as much as it's an awful job, I encourage people to do it, because right now there's just not enough caregivers for all the people that are super old right now and unable to take care of themselves, and they're unhappy and miserable and stuff like that. And they'll laugh at anybody who says something about aging gracefully.
Right. So I mean, this is the silver tsunami that's sometimes referred to. That the baby boomers are getting old, and it seems as though there's unless there's some form of rejuvenation therapy or something else out there that helps them sort of stay fit for longer, extend their health span, there will be a huge burden on the medical establishment.
Yeah.
Is that correct?
The silver tsunami is going to be a major problem in the future if we don't figure out a way to, what my expression is, cure aging.
Well, since we're on the topic of curing aging, now you wrote that book. Do you want to give us a rundown of what's really in it? And people should know that it's about telomeres, and it's about trying to rejuvenate telomeres or reduce the speed at which they shorten. I may be a bit off there, but do you want to give us some background there? What sort of insights in the book that might be counterintuitive?
The inspiration for the book was I give a lot of presentations to general audiences, medical audiences, scientific audiences, things like that, about the thing, and then I get a lot of questions afterwards. Okay, about what's going on with so the book is pretty much answering all the questions that people ask. But it includes the basics of what telomere biology is all about. It explains it in a very non-scientific way using bricklaying and other things as models, factories and things like that as models to explain what's going on. But the bottom line is that when we are first conceived, the tips of our chromosomes are 15,000 bases in length. Bases are a unit of measurement in DNA. A DNA is typically about 100 million bases in length. The telomeres at the very tips are 5 million bases, 15,000 bases in length. And think of a long shoelace, essentially, where the genes are organized along the shoelace, where the caps on your shoelaces are equivalent to the caps on our chromosomes, which are called telomeres. And we know when the caps on our shoelaces get short, our shoelace falls apart. And the same thing happens when our telomeres get short, our chromosomes start falling apart and cause all kinds of diseases and mutations and increases our chances of cancer and all that kind of stuff. So we want to keep telomeres long. When we're first conceived, our telomeres are 15,000 bases. And then every single time a cell divides, each and every time a cell divides, the telomeres get a little bit shorter. And they get shorter not because something's chewing them away. They get shorter because our cells lack the ability to duplicate a chromosome all the way to the very tip. So every time a cell divides, you have a cell and it's going to divide to become two daughter cells. Everything inside that first cell the parent cell has to be duplicated so that the two new daughter cells has everything that parent cell had. And that includes the DNA. The DNA that makes up our chromosomes has to be duplicated. When the enzymes called DNA polymerase I come to the very ends of the DNA during the replication process, it can't replicate the very end. I compare this to bricklaying, laying a whole new row of bricks on top of a brick wall, with the bricklayer standing on top of the wall, stepping backward each time. And when the bricklayer reaches the very end of the wall, he steps backward, he can't lay a brick in the last place he was standing because he's fallen off. So as a result, the new chromosome or the new row of bricks is shorter than the previous one. And so that's why our telomeres get shorter. Now there's things we can do to accelerate that, but if you lead the perfect genetics and have the perfect lifestyle, your telomeres are shortened at a rate that you can live to be theoretically 125, but not longer than that. Now, in our reproductive cells, that same process is occurring, but we have another enzyme called telomerase that's like in the bricklayer, it would be an angel that flies in and puts that brick in place, because the angel has wings, can move around, whereas the bricklayer is kind of pretty much attached to that wall. So the angel puts that last brick in, so that makes the new row of bricks as long as the old row, just like the new chromosome now will be just as long as the old chromosome. And we have found that if we increase the concentration of that angel, or increase the concentration of the enzyme telomerase beyond what we find in our germline cells, our reproductive cells, we find that they can actually lengthen the bricks, or the DNA, so telomeres. So as a result, it'd be the same as making a new row of bricks was actually extending in both directions longer than the old row of bricks. And that's what causes age reversal, okay? When the telomeres get longer. That's what Dr. Ron DePinto did with the mice. And that's what I think we're a year away from doing with a small molecule drug, and we can do right now with the gene therapy.
So it's, I mean, like you and Aubrey de Grey seem to agree on the major goal of ending aging or defeating aging. Now, in general, okay, whether you believe that aging is a disease or not, the issue is the diseases associated with old age need to go, or they need to be forestalled. Buying more, like telomere therapy might buy more time. It's like one of the shortest fuses, as you described, in the many sticks of dynamite that we have ready to go off in our bodies. But you and Aubrey do disagree on whether like a telomere lengthening will help. Now he believes that telomere lengthening may actually increase the risk of cancer, and he also thinks that there are other cells in the body which need to die and which don't replicate at all or anywhere near as fast as some others. Why do you disagree with Aubrey, and what counts do you disagree, and where do you think is wrong?
Well, on the second point, we don't disagree at all, and that's that we do want to get rid of senescent cells in our body, okay? So he's working on approaches, and so are other labs working on approaches to kill off, to cause senescent cells to die. The problem with senescent cells, they don't die, they just sit there and do the wrong thing and survive, okay? So if you could kill off the senescent cells, that would be beneficial. At least we've been able to show that in, or not we, but scientists in general have been able to show that now in mice. Now, let's say it's true in humans. Well, wouldn't it be better just to not let the cells get senescent to begin with? And that's my approach. So my approach, so yeah, we don't want senescent cells. Either get rid of them or don't let them get produced. I'm working on preventing them from being produced, where Aubrey's working on trying to get rid of them. Okay, so, but yeah, senescent cells are not good for you. Senescent cells mean that they're really old and do nothing except communicate with other cells and make them sicker, and it's just a bad thing you don't want. Now, on the first question about the will lengthening telomeres or inducing telomerase cause cancer, there were studies a long time ago suggesting that. I think most of the studies have been since disproven. Their techniques, the delivery techniques, not the actual enzyme telomerase, was actually causing the cancer. But there hasn't been a paper since like 1995 that suggested that telomerase causes cancer. There's a few papers out right now that say that long telomeres increase like glioblastomas and things like that. But telomere length measurement is so difficult to measure right now. It's not the science that we'd like it to be yet, and so it can give bad results a long time. But the key is that for every paper that's saying long telomeres cause cancer, there's 100 that says short telomeres cause cancer. And so we do have this kind of like, is the right word, dichotomy, or something like where we have two different choices. One is telomerase causes cancer or lack of telomerase causes cancer. Okay, short telomeres cause cancer or long telomeres cause cancer. We have that both kind of thing, those questions. We have papers in scientifically peer-reviewed journal articles right now saying that both are a possibility. But the number of papers that are suggesting that short telomeres cause cancer and the lack of telomerase causes cancer are overwhelmingly better than the other ones. And it also not just.
Well, why is it? Why is it that short telomeres cause cancer, do you think? I mean, I think I've heard you say one thing, and that is that short telomeres can cause, or can correlate with a mutation in the cell, which then causes overexpression of telomerase. So and that goes along with you know, what cancer ends up doing, it does try to protect itself seemingly by increasing telomere length through expression of telomerase.
Something different, but what you said is generally correct. But you skipped a few steps. Okay, so
yeah,
what happens when telomeres get really short, like in senescent cells? The chromosomes become unstable, just like your shoelaces become unstable when the caps get really short. The chromosomes start breaking and rejoining, breaking and rejoining. You get chromosome rearrangements, you get mutation. These big chromosome rearrangements you can see in the microscope. Okay, there's also point mutations that are too small to see in the microscope, but these all cause significant changes to the cell. And when they start doing things like causing the cell to lose growth control, then that's when your cell becomes a cancer. Now, then the telomeres even get shorter, which increases the mutation rate and stuff even further. And so most of those cancers will die, but because the mutations rate's so high, you're going to find some cancers will still survive, and they do it by inducing telomerase, by creating a mutation that induces telomerase. Or they induce a mutation that causes what's called the alt pathway, alternative lengthening of telomeres, which is more a recombination method than telomerase. But either way, 85% of all cancers that have survived big enough. Okay, so for instance, if you had a cell in your body suddenly lose growth control, before it got to the size of even a mole on your face, it will have died out because of the telomere shortening. But of course, we all know that a lot of moles end up mutating to become cancer. Okay, and that's because the high mutation rate that's going on there to induce telomerase, and as a result, that makes that cancer an immortal cancer, and so it can spread. So telomerase didn't cause the cancer caused telomerase. Okay, so the telomerase comes second. So you get a cancer, and then you get an immortal cancer. If we could keep the telomeres from shortening to begin with, that would prevent the telomerase from being induced, because it would prevent mutations that induce it. And it would also prevent the mutations that cause the cell to lose growth control. At the same time, you've got your telomeres shortening in your immune cells. And one of our best ways to fight cancer is our immune system. Our immune system is extremely efficient at fighting cancers. It's just that a lot of times people get cancer, that the cancers overwhelm the immune system, and sometimes that's because the immune system's telomeres got shorter, and so your immune cells are senescing. So keeping telomeres long in your immune cells will decrease the risk of getting cancer, because it increases the immune system's ability to fight it, and it also helps you fight it if you have it. So short telomeres. So as I said, short telomeres can cause mutations that lead to cancer, and then the more mutations lead to telomerase production. That explains, for instance, why most cancers have critically short telomeres, because the telomeres got short first, and then additional cell division made them even shorter. But yeah, the bottom line is that if you have a choice between no telomerase and telomerase, I think the no telomerase is going to decrease your risk of cancer. No, it's going to increase your risk of cancer more than telomerase will. And you don't have a third choice. You know, it's you either have telomerase or you don't have telomerase. There is no third choice. And so we have to choose one of those things. But I believe look at it. We get cancer all the time now, okay, without producing telomerase in our cells. I believe that producing telomerase in our cells is going to decrease the incidence of cancer, not increase it. What Aubrey and I do agree on is that we will not know for certain until we do something. So Aubrey and I both support each other, that I want to see his research get done. He wants to see my research get done. And we want to then answer those questions, okay? So we don't know the answer. It's just guessing, best guessing right now. His best guess is different than my best guess. I do want to add that my award, second place for National Inventor of the Year in the United States, was for my cancer research. I was a cancer researcher.
You know a few thing or two about cancer. That's good to hear. Yeah.
It wasn't about my anti-aging research at all, but it was because it was related to telomeres and telomerase and the roles of cancer. That's why it's still something I mentioned is involved in telomere biology, but it wasn't for my anti-aging research at all.
Sure.
I do believe that we are going to see this, but I'm a scientist. I don't go with guessing and things like that. I say I wouldn't be surprised if we see a decreased incidence of cancer, but we're not going to know until we have a drug that lengthens telomeres and see what happens. And Aubrey and I are both looking forward to seeing what happens. And I strongly believe, and you can ask Aubrey when we do our next talk in a few days, if he agrees that, because in The Immortalist, he clearly says, I say the same thing, is that we won't know until we try. And that's what we got to do. And since Aubrey and I are both suffering from a disease called aging, neither one of us are going to be upset if somebody else finds a cure for our disease. So we support each other, and we both support a whole lot of other people working on aging too. It's one of the most collaborative fields I've ever seen.
Absolutely. So, let's get back to the metaphor of the many sticks of dynamite that are sort of, with different lengths of fuses. You're suggesting that once we, fix the telomere and telomerase induction, then we'll have to work on the next problem. What do you imagine? What are the next problems that need to be fixed?
Oxidative stress. Oxidative stress, mitochondrial dysfunction, advanced glycation end products, SIRT1, all these things, a lot of these things, there's a good chance would disappear when we lengthen the telomeres. Another study by Ron DePinel showed that keeping telomeres long actually prevented oxidative stress and mitochondrial dysfunction. And I believe that a lot of the studies shown it looking at like SIRT1 decrease, human growth hormone decrease, things like that, what is it, CD28 negative T cell decrease, things like that. Or is it increase? I can't remember. But I believe that there's a good chance we might find when we keep telomeres long, those things don't happen. So we might, and Ron DePinel in an interview one time said that he thought that telomeres were the kingpin of aging. I should tell you that Ron DePinel is a cancer scientist, and his main goal was to find a way to inhibit telomerase to cure cancer, or to at least study. So a lot of his work was done to show that keeping telomeres or inducing telomerase was going to cause cancer, and he found the exact opposite. And maybe that's why he went off to MD Anderson to become a cancer researcher, but or to actually head the whole thing. But I don't know what's going on there in terms of him, but I believe his data, even though he was trying to show the exact opposite, is showing that telomerase actually decreases the risk of cancer. And I should say, I don't think he was trying to show anything. Ron DePinel is one of the best scientists I've ever met, and he just wanted to ask the questions and got his answer. So without actually trying to bias the results one way or another. But now I forgot what the last question was you had. What was it? We were talking about sticks of dynamite.
Yeah, sticks of dynamite.
Leonard Hayflick, the person who's discovered the Hayflick limit, he published a really good book on aging, and in that he describes all of the theories on why we age. And so I like to show a slide when I speak about all these different theories, and I don't try to say one theory is true and another not. I just say they're all true. We have a whole lot of things happening to us that causes us to age. But I think of them as each being a stick of dynamite that's burning inside of our cells. And so which stick of dynamite has the shortest fuse? And that's the one that we need to focus on first. In humans, I think it's telomeres. Telomeres are our stick of dynamite with the shortest fuse. Now, in mice, their fuses are going to be different. Their stick of dynamite with the shortest fuse is oxidative stress and mitochondrial dysfunction. And maybe someday if we cure that in mice, they'll maybe live to be 90 years old and have telomere shortening, die from telomere shortening. Now I've noticed your picture is frozen. Can you still hear me?
I can still hear you, and the picture that's just frozen looks rather embarrassing.
That's okay. But I believe that because we do know that oxidative stress and mitochondrial dysfunction can affect humans, especially in smokers and obese people, I believe that if we do stop telomere shortening, we will still have oxidative stress and mitochondrial dysfunction to some level. But I believe that that'll give me 40 more years to find a way of fighting that if nobody else has beforehand. Because there's a lot of really good scientists working on trying to do some of those things right now. And I hope them all the best, because wouldn't it be great if we solved all the different possible, solved ways of putting out all those sticks of dynamite at once. And then if we don't, or if we put them all out and we still have problems, then I'm just really grateful to the people that are working on cryonics, however you want to pronounce it, or brain uploading and things like that, because those are the last resorts. And I'm excited about the 21st Century company with Greg Fay as the chief scientific officer. They've really revolutionary breakthroughs in cryonics recently.
That's right. Yes, and I just recently interviewed Kenneth Hayworth, which you might know, and he sort of ran the brain preservation prize for small animals. What's interesting, it wasn't such a small animal which ended up being presented for the prize. They could have given it like a rat brain or a mouse brain. Instead, they did it for rabbit brain, which is sort of mid-size.
Well, I think it's because they've already done mice. Okay.
Yes.
And mice were small enough that it did work. You know, I'm not going to kind of pretend that I understand everything they're doing, and I don't want to, because I feel that they're really good hands. Ken and Greg are just they're the people that really are going to save us all. But I think the major problem is not the freezing, but it's the thawing, because trying to prevent ice crystals from piercing and breaking open the cells is the problem when you're doing the thawing. And they could do that with mice because the mice brains were smaller, but it was harder to do it. And I think accomplishing this in the rabbit brain is really a major accomplishment. I think it's not long before we can do this in whole humans.
Yeah, that's right.
I hope
we would be able to.
Yeah.
I think the actual solution which they use to replace all the water in the brain and stop ice crystals forming is actually deadly if someone were to put that in their system while they were alive. So it might be quite a long time until we work out a way to reverse the damage that does, or remove that while also thawing out a brain. He suggests that it's more likely people will be uploaded, but I don't know. That's a long way off. It's highly speculative. Yeah. So one of the other things I'd like to ask you is, so yes, this Hayflick limit, do you think that, like, well, we're talking about the Hayflick limit, but each time we have a breakthrough, like if there is a breakthrough in increasing telomere length and inducing telomerase, then that buys time. And Aubrey sort of suggests that we, like, buying time is kind of like a sort of an escape velocity. We get a bit more time until the researchers, the scientists, the engineers come out with the next therapies that'll keep us alive for longer. For those who are alive today, that's important, right? But we all understand that the ultimate goal needs to be to fix aging completely. But even if we fix it a little bit, an extra 15 years for someone means an extra 15 years of research coming down the pipeline and perhaps resulting in the next levels of therapies that they can take. And that's important for people listening who are alive today.
One of my favorite quotes of all time was from Dr. Terry Grossman, who's also in the Immortalists, and that's "live long enough to live forever." And that's exactly what it's about. It's the bridges, crossing these bridges so that you can stay ahead of that threshold that's knocking us off, and be around for every time there's a new cure coming along. There's a big conference coming up that I think is going to be the biggest conference in this field of all time. So never is going to there been anything this major in the field of anti-transhumanist stuff as this conference. It's called the RAD Festival, RAAD.
Yeah, I've seen that.
I'm a speaker. Aubrey's a speaker. speaker at that. Liz Parrish is a speaker at that. There's a whole bunch of speakers. Ray Kurzweil is a speaker. I think maybe even Dr. Terry Grossman's a speaker. I can't remember or not. But I think if anybody, and Greg Fay is a speaker, people want to know about all the different things that they can do to extend their lifespan and cure their aging. RAAD stands for Revolution Against Aging and Death. Okay, so it's not just aging, it's everything that knocks us off. It's called the RAD Festival. If people just go to www.raadfest.com, they can sign up for it. And I'll tell you, there's early registration for that. It expires in about less than a week from now, actually about 4 days from now. And so people should go there fast. But the conference is actually going to be August 4th through 7th. It's going to be in San Diego. And this is, I'm calling this the Woodstock of biotech and aging. Okay, this is going to be a major, something like 1,000 people have already signed up for it. And it's not even, 6 more months before it actually takes place.
Wow, so, incredible.
I think this is going to be the biggest event of all time in our field. So people should listen and learn a lot about what we're doing.
Absolutely. It sounds like a place to be. Yeah, I'm in Australia. I can't tell yet whether I'll be able to make it over there. So if I do come, I'll probably be paying the full price.
But, a lot of my friends from Australia, New Zealand, Hong Kong, China, South Korea, Japan, South America. I'm trying to think of where I've got other friends. Dubai. A lot of them are coming to this festival because they know it's going to be the biggest thing of all time.
Definitely. Well, yeah, I guess.
Did I lose you? Okay. I cannot hear you. I cannot see any motion at all. So if people can hear me out there, that's really great. But I don't know if anybody out there knows if they can hear me or can let me know if they can hear me. I guess I'll just wait to see what happens. Okay, so Adam just texted me saying that he can hear me, and I can't hear him. I can't see him. So, Adam, if you want to text me some kind of question you would like me to address, otherwise, okay, so have you heard about Google DeepMind Health? No, I have not. DeepMind Health is, therefore, since I haven't heard of it, there's nothing I can say about it. But, you know, let me just keep talking, assuming people can hear, and I'll wait for more questions. Let's see. And Gary Anderson, who I am glad to see is on the call, he just texted me. Okay, he said, "We hear me, just talk." Okay, so one thing that I think is really exciting about the telomere field is that there are animals on this planet that have no detectable aging. And it turns out in all cases, it's because they don't have any telomere shortening. They have telomerase produced in all their cells, not just their reproductive cells. They have no detectable aging. They have no telomere shortening, and they rarely get other diseases. And I want us to be like these animals. And the first one that was discovered was actually lobsters. Lobsters, you know, people don't find old lobsters. They find unhealthy lobsters sometimes, but they don't find old lobsters. Lobsters just keep on growing and growing. They found a whale that had a harpoon in it, a humpback whale, and they carbon dated that harpoon because it looked kind of old, and it was 130 years old. So the whale was older than 130. Now you would think that people would know this kind of stuff, but people really didn't start measuring how old animals live until around the time of Darwin. And there is, in most cases, animals don't have like rings on a tree where you can just count the rings to find out how old they are. To find out how old they are, you have to really be there when they're born and keep them in captivity in a cage or an aquarium and just watch. Well, lo and behold, people have been doing that and find lobsters are living 125, 150 years and still not showing any signs of aging. Tortoises, like Charles Darwin, he had a pet tortoise named Harriet. She just died recently at 180 years old, long after he died. And then there's other tortoises that have been documented to be 200 years old. But the way they know is because they were there when the animals were born. Now, the Harriet did not die of aging. Theoretically, she probably died from some virus infection of some kind. But there's other animals that do have rings on a tree, like clams. Clams have no detectable aging. They have telomerase produced in all their cells. But clams produce a new stripe on their shell every year. So you can catch a clam, count all the stripes, and determine how old that clam is. And now people have found clams over 500 years old. In fact, there was one recently announced. It was 507 years old. They named the clam Ming. It was a female. And it was named Ming because it was born during the Ming Dynasty. I think that's real exciting. I want us to be all like those animals, except I don't want us to look like them. But I really believe, and let's say I wouldn't be surprised if in the next few years we find people start getting younger and healthier. And I'd like to see that happen with a lot of people. I recently got a chance to meet a lot of the people from the 1960 and 1964 Olympics, the gold medalists, and they're all super athletic still, very good shape. Dick Fosbury is one of my favorites because I used to be the high jumper and do the Fosbury flop and stuff like that. And I would love to see him and Eddie Hart and Ray Norton, and I'm forgetting all their names. I'd love to see them all get treatment to bring them back to their Olympic selves again. Let's see, I just got another question. Can I discuss definitions of immortality, biological, and other religious? I'm not a semantics person. I don't know what the definition of immortality is. My definition of immortality is like Greek gods. Greek gods can't be killed. They could be sliced in half, but they'd still be alive. So that's my definition of immortality. But science, in the biological sense, we do have this definition of immortality. I don't know where it came from, but when you take human cells or any type of animal cells and you grow them in a petri dish, if they just keep growing and growing and growing and never reach senescence, we call that immortal. Of course, you can still add hydrochloric acid to those cells and destroy them, but that's not how the definition of immortal is used there. In the religious sense of immortality, again, I guess that's the Greek gods, because it was Greek mythology, which I guess is considered a religion. But the definition of immortal is whatever people want it to be. I don't really care. I'm not a semanticist. I don't even know if that's a word, to tell you the truth, but I don't really get into what is the definition of aging. People ask me that all the time. What is aging? What is aging? And then they'll say, what does curing aging mean? And it's really frustrating to me because I don't know. When I was 10 years old and my father first put the idea in my head, he said, Bill, when you grow up, become a doctor and cure aging. I didn't ask him what he meant. I knew exactly what he meant. And like when I give talks and stuff like that, I like to show pictures of Betty White when she was in her 20s and Betty White now. And I just point blank ask the audience, is there anybody in this audience that doesn't know which photo of Betty White was taken first? Because if they do, then they know what aging is, even though they can't necessarily put it in words. But people want to ask that question all the time. And then what does curing aging mean? Well, curing aging means if we could turn Betty White back into her younger self, both physically and health-wise. So, semantics, I just don't get into that. I'm going to lengthen telomeres, and I believe that it's going to benefit humans in one way or another, and so it's still worth doing. I got another question here. Will bodybuilding help? And that's HIIT, and I got to confess I don't know what that means. What are some economists' approaches to extending health spans? Oh, high interval intensity training. I believe high interval intensity training is extremely good for you. I do this all the time, even though I'm an endurance runner. I actually will confess that I think high interval intensity training is a lot better for you than endurance running, health-wise. But I do endurance running because of the adventure. I'm just obsessed with it. But I do combine it with high intensity and high interval intensity training. And I got to thank Dr. Al Sears, who's one of the doctors in the world that first has recognized the importance of telomeres, for convincing me that high interval intensity training is important. I've been doing that ever since. Going back, even though I do say that endurance training, exercises, and stuff like that do help keep the telomeres from shorting, I do believe that bodybuilding and things that induce muscle or cell division. Okay, so when you're lifting weights, you're actually tearing your muscles apart, and then the bulk muscles come from actually new cell division that fills in those gaps. Well, by definition, every time a cell divides, the telomeres get a little shorter. So I'm worried that bodybuilding might actually accelerate the aging process. I believe that microdermabrasion also accelerates the aging process for the same reason, because when you do microdermabrasion, you're killing fibroblast cells in your skin and causing other cells to divide to replace those. And because of the cell division and overabundance of new cells, it makes you look younger. But I think it's actually making you older, or those skin cells older, because the telomeres are getting shorter. I'm going to say one other example, and that's human growth hormone. I think the human growth hormone will accelerate telomere shortening and therefore aging, because it induces bone cell and muscle cell proliferation or cell division. But all these things are good for you, I mean, they do make you look good and feel good, and isn't that what we're all here for? I always say, what's the point of living a long time if we're not living? So I highly encourage the bodybuilding. I think bodybuilders love the results that it has. I think people who are doing HGH love the results they're getting. People doing the microdermabrasion, they love the results they're getting. And I'd say do it. Don't worry, because if it is causing accelerated telomere shortening, we'll have a fix for it before you have any problems. I'm looking to see if there's anything else here. Now, I did get a text again from Gary. No, it's actually not from Gary. It is from his partner, Don Hargreaves. Why are you chicken of Liz's virus approach? That's a good question. Yes, I got to say I am so impressed with Liz Parrish for doing this. I think this is answering all kinds of questions for the scientific community that we need to know the answers. But it's just I want to be one of those people that benefit from all the research and studies. I don't want to be one of the actual casualties from the research. And there's always that possibility. And because the gene therapy approach has not been approved by the FDA and stuff like that, I am a little concerned that it might have side effects, not because of telomerase. I believe telomerase has been shown over and over and over again to have no health effects, negative health effects on human cells in petri dishes or elsewhere. But I believe that there's always a chance that the delivery method or something like that might cause some problems, even though the virus that Liz used has been shown in other clinical studies with other genes to actually not have any of these other side effects. I just got to wait till 100,000 other people have tested it and shown no side effects, and then I'll do it too. But I don't want to do all this hard research that I'm doing and find out that I end up dying as a result of the research. Liz Parrish is a lot more concerned, I guess, about other people. I'm concerned about other people too, but I do put myself first. I want to make certain that I'm there and healthy and stuff like that to enjoy the future life we have. And there's a term that I use. I learned a long time ago from a guy named John Vigil who lives in Las Vegas. It's called FOMO, F-O-M-O, and it stands for fear of missing out. I don't want to miss out. When we find life on other planets and stuff like that, I want to be here. I mean, Liz took a chance of risking the possibility that she might not be around, and I'm glad she still is around, but I'm also glad that she did it, and we we're learning from her experiences right now.
Can you hear me now?
Yes. Hey, I can hear you.
Hey, fantastic.
Now I thought our call was only going to be 30 minutes, and now we've gone an hour, 18 minutes. But
yes.
I want to listen.
Well, if there's any more questions that the audience wants to answer, I wouldn't have thought to ask that question that one of your friends just reasonably answered. Have you done a Reddit AMA yet?
Well, repeat that. What is that?
Have you done a Reddit Ask Me Anything before?
I've never heard that.
Okay.
I always answer every question. Whenever I speak at conferences, I always say, I'm standing around the last person, please. I want to make sure nobody feels like they didn't get an opportunity to ask me questions. So yeah, if people can ask questions now, they can go for it. And whatever this Redis thing is, are you going to explain that?
Reddit, yeah. So sometimes Reddit is a common forum, a community of people who like to talk about all things under the sun. And certainly anti-aging or health span increasing is one of those. Now, Aubrey de Grey, and I think Liz Parrish has as well, they often, in Reddit, they target a specific expert and get them to basically answer all the questions that people who are on Reddit, who are participating in the Reddit forum, they'll answer all their questions. And yeah, I was just wondering whether you would consider doing one of those.
Absolutely. I want to do that. That sounds like a lot of fun. I just send me the information on how to sign up for that. I'll get on that instantly.
Yeah, I think that'd be quite high impact. I can't see any questions that people have been asking at this point in time, but are there any, like, if people were to take something away from listening or watching this video, what would they be? What would be the major points that you'd like for them to take away?
I guess the major point that I'd want to make is that there's a lot of great science going on in this world by basic research labs. There's people that have the cure for everything: AIDS, herpes, cancer, Alzheimer's, all that kind of stuff. But they don't have funding. But they don't have the funding to complete their research. And so we keep seeing these announcements about somebody doing some experiment that shows that they can cure some disease, and then you never hear them anymore because they don't have funding. And the world thinks everybody has funding and that it's all going to happen. But I keep saying aging is not going to cure itself. Okay, Aubrey de Grey, Liz Parrish, me, everybody else that's in the anti-aging world with great ideas and the anti-cancer ideas and the other diseases, we all need funding to get the research done. And so please quit thinking that we all have funding. The research isn't getting done because we don't know the science. The research will get done because we do know the science if we had the funding. So that's the main message that I want to give. And the other message is that, hey, the world's going to change really quick. We are close. We are very close to curing aging.
And that's something that can, it can benefit everybody potentially. So what can people do? I mean, what sort of things can, like, an average person, with an average income do to help the cause?
Just spread the word. And if somebody's sitting on an airplane sitting next to a billionaire who's saying, boy, I wish somebody would cure my aging, tell them about me. Tell them about Aubrey. Tell them about Liz Parrish. And I apologize. There's other people out there: Mike West, Bryant Viaponto, Mike Fosso. There's a lot of people out there working to do like that. And I'm not the only company either. I want all these companies to find Genescient, for instance. I'm sorry I can't remember all their names right now, but there's a lot going on. Biotime is another company. Everybody needs funding to get the research done. Let's find ways to get it. What else can they do to help? They could start.
Yeah. I think Genescient is run by Gregory Benford, who came to speak in Australia in year 2011, I think, or 10. Yeah, when the science fiction convention was on. He spoke at a conference I organized.
Other things that people can do is help remove some of the myths that exist. Okay, so like, let me take the telomere field in specific here. As we already discussed earlier, there's a lot of people that think that telomerase will cause cancer, and I've explained that I believe that the lack of telomerase is more likely to cause cancer, which we already have. But if people could start learning about this stuff, and that's one of the reasons I wrote my book, Curing Aging, and it's available on Amazon.com. And and if somebody in a foreign country can't get it, send me their address. I'll make certain they can get it, some one way or another. But read the book. It's written for the non-scientists. Learn the truth so that a lot of these myths and bad things being said about the field can be dismissed. And that's going to increase people's awareness that this is something important, and therefore investments will start occurring. Or, right now I'm not even looking for investors anymore. I'm just licensing some of our discoveries to make products that are on the market already, and more coming in the future, that we're going to get our revenue, our funding for research from the royalties. And so one thing you can do is buy these products and help others buy these products, and get out there. These companies are looking for distributors now. Help them sell the products. You make a lot of money, they make a lot of money. It's those kind of things. You're actually contributing to the cure for aging when you do that. I could go on and on with key messaging. The funding is the key one.
Yeah, it'd be great to see some well endowed financially people fan some money in your direction, Aubrey's, Liz's, Gregory Benford's, and many of the others that you mentioned before. But yeah, look, I feel as though we've really covered a lot of ground. There's so much more that we could cover. But I'd just like to remind viewers and listeners that there will be an online panel in a few days with Bill Andrews, Aubrey de Grey, and Liz Parrish there. So, well, in some parts of the world, that'll appear on the 29th. In other parts of the world, it'll be the 1st of May. So, yeah, that'll be a future day celebration.
It'll be the 1st of March, Ron.
Sorry, 1st of March. What am I talking about? Yep. All right. Well, it's been fun.
But the other thing I want to re-say is go to the RAD Festival, okay?
Yes.
RAADFEST.com. Okay, this is going to be the most exciting thing. You're going to hear everybody who's anybody in the field talking and about what's going on. So.
Yeah, fantastic. Yeah, and I think the organizers there have been organizing a number of longevity events over the years, so they seem to know what they're doing. People
Unlimited, which is a major organization run by Jim Stroh and Bernadine. There's also Life Extension, Bill Falloon, who's one of the key promoters of that, David Kekich. I mean, there are so many great people in the field of anti-aging that are just as passionate as I am about doing it. And they're all going to be there, and they're all putting this thing on. I feel like I'm helping my best, but I'm not doing nearly the job that Bill Falloon and Jim Stroh are doing, and David Kekich are doing. But this is going to be a monumental event for anti-aging and transhumanists.
Definitely. Well, if there's if anybody can, it's a good time to buy those tickets before the early bird specials end. Yeah, I'm in Australia. As I said, I don't know whether I can actually make that date at this time. So, but in any case, I'll endeavour to get over there, and hopefully, I'll be able to get a lot of more interviews. That'll be awesome.
I hope I see.
Yeah, definitely. So, well, if there's any concluding remarks that you'd like to say, and then we can, end the interview.
Yeah, nothing that comes to mind right now. I've said so much, but it says people accept that people should listen in a few days, as you just said, to when me and Liz and Aubrey are speaking, and you might hear a lot of me say the same things I've already said today, but hopefully that discussion will come up with new subjects.
Yeah, absolutely. So it's been fantastic having you, Bill, and I really look forward to the future and see what your research turns out, as much as I do Aubrey's, Liz's, Gregory Benford's, and many of the others. We're living in exciting times, folks. Keep your eye on this, and if you got some money, donate it in their direction. It'd be awesome. So thanks very much for your time. I really appreciate it.
Thank you. Well, goodbye, everybody.
See ya. And thanks everybody for listening. Please subscribe to the channel, and yeah, I'll look forward to doing another interview and letting all you guys know about it soon. Bye.